Hi guys! So I am Sheila Castillon and I will be your medtech/mentor for today. So I will be performing right now the manual method of infiltration to our clear tissue specimen. So in this step, we will be removing the clearing agent and we will replace it with a medium, which is commonly our paraffin wax, and this wax will completely fill all tissue cavities. So the question is, why does the tissue need to undergo infiltration? So this is done so that the tissue will have firm consistency, which will allow us later to have easier handling and cutting of thin sections without causing any damage to the tissue and its cellular components. So, shall we start the infiltration process now? First, we will gather all of our materials. So we have here our paraffin wax, our paraffin dispenser, the 3 glass dishes, clear tissue samples, which should not be more than 4 millimeters in thickness. We have here the string, gauze, stick, paper towel, and lastly, we have here our paraffin oven. Next, we will melt our paraffin wax by heating it 2 to 5 degrees Celsius above its melting point. So the most commonly used type of paraffin wax has a melting point of 56 degrees Celsius. Remember also that the best way to determine the temperature in melting the wax is between 55 to 60 degrees Celsius because beyond 60 degrees Celsius will cause harmful effects to the section. Remember, wala ta nagaluto o tissue diri guys, okay? While heating the wax, we will prepare the setup for our infiltration. We will wrap our tissue specimen with a gauze and tie it with a string. We will tie the other end of the string to a stick so that we can easily transfer it later to the next glass dish. Now we will label also the tissue specimen using a pencil. Now that the paraffin wax has already melted, let us now proceed to the next step. Next, label and fill the 3 glass dishes with paraffin wax. We need to immerse our tissue sample for 30 minutes in the first paraffin wax bath, another 30 minutes in the second paraffin wax bath, and 45 minutes in the third paraffin wax bath. This is done for us to be able to ensure the complete removal of the clearing agent and the infiltration of the tissue with the paraffin wax. Then, remove the tissue sample from the last paraffin wax bath and place them on paper towels. Finally, we will now prepare the tissue so we can now proceed to embedding.
I am Trisha Marie Junio, your medtech for today. After the infiltration process, we will now proceed to the next process called embedding, which is also known as casting or blocking. This histopathological technique involves the placement of the tissue in an embedding medium which will be allowed to solidify. Without further ado, let's start our embedding process. These are the materials needed for our activity.
First, we will need the embedding medium, which is a melted wax. Next, the impregnated specimen. The 4 steps to guide us when we are orienting our specimen. The embedding mold, which is a disposable paper boat. And lastly, a cold water where we will cool down the temperature of our wax. Let us now proceed to the procedure proper. For the first step, prepare your embedding molds. Do not forget that we have to use 1 mold per tissue. Remember, attend to 1 sample at a time because you might interchange your samples. For the second step, dispense a small amount of wax to act as a base. Immediately transfer the tissue into the central portion of the mold. Use your forceps to guide you. Observe proper tissue orientation because this will be reflected in your tissue sections. Third step. After placing the specimen, fill up the rest of the paper boat with wax. Remember, in most cases, whatever medium is used to infiltrate the tissue is also the same medium that will be used to embed the tissue. Fourth step. Immerse the paper boat with the tissue immediately in a cold water to cool down the temperature of the wax. Fifth step. Once done cooling, detach the block from the paper boat and don't forget to label it. You will determine if the tissue block is ready once it is easy to peel off from the embedding medium. And finally, we now have our tissue blocks.
So why is embedding important in the first place? Embedding is important because this process allows the hardening of the tissues, giving them firm consistency and better support, thereby facilitating the cutting of the sections. I hope you enjoyed doing this activity with me. That would be all for our embedding process. Thank you so much and have a nice day.
Good day, I am Lucille Therese G. Mordeno and I am your medical technologist for today, and I am going to perform floating out and fishing out of tissue specimen, or simply known as fishing out in the laboratory. Is a technique used to mount a tissue specimen into the glass slide, and it usually performed after the tissue specimen was sectioned. And here are the materials that we are going to use. And here are our materials. We have our water bath. The temperature must be within 42 degrees Celsius. It is because high temperature of the water bath most likely cook the tissue specimen. We have our tissue specimen. The tissue specimen will be placing the water bath immediately. And we have our adhesive. The purpose of the adhesive is to have the tissue specimen to mount in the glass slide properly. We have our cotton swabs, tweezer, and our clean glass slide, which was washed in 95% ethanol. And we have our pencil for labeling, our paper towels for cleaning and drying for tissue specimen. So the first step is to wash the glass slide into 95% ethanol, and you must make sure that it is completely dry. The next step is label it using the pencil. Remember, you must not use a ball pen. It is because it can affect in the staining process. The next step is to add the adhesive into thin layer into the glass slide. Then fish out the specimen into the water bath. Okay, and then you must completely dry out in some paper towels so that the tissue will not be disrupted. And that's how you mount the tissue specimen into the glass slide. And that's how you perform floating out and fishing out the tissue specimen.
everyone, I am your histotechnologist of the day, Joshua Sales. Today, follow me as I perform the H&E staining process. Before we start the staining process, let me first define what is staining and the H&E staining. Staining is the process of applying dyes on the sections to see and study the architectural pattern of the tissues. The methoxylin in eosin, or commonly known as H&E stain, is the most widely used staining technique in histopathology. The combination of the two dyes stain various tissue elements and make them easy for observation. In our H&E staining, the first step is to immerse the slide in xylene. This is done in 2 changes of xylene. Each immersion should last exactly 5 minutes. The second and next step is to dip the slide 10 times in alcohol formol. In the third step, we also dip the slide 10 times in 95% ethanol with 3 changes. The fourth step in the process is to wash the slides in soapy water. Then, rinse the slide well with clean water and drain excess water on paper towels. The fifth step is to stain the slide in hematoxylin for a duration of 5 to 10 minutes. Hematoxylin is a basic dye, thus it stains basophilic structures like the nucleus of the cell purplish blue. Drain the excess stain and rinse with water. The sixth step after the hematoxylin staining is to decolorize sections in acid alcohol with 2 quick dips. The purpose of decolorization is to get rid of the irrelevant parts of a tissue that had stained such dye or reagent. This is for the clear differentiation of the dye-specific part of a tissue or cell. Rinse the slides immediately in water. The seventh step is to immerse the slides in ammonia water until the tissue turns blue with an average duration of 5 minutes. Rinse again in water and drain the excess on paper towels. The eighth step in the process is to stain the slide with eosin Y for 1.5 to 3 minutes. Eosin is an acidic dye, thus it stains acidophilic structures like the cytoplasm of cells red or pink. Drain the excess stain and rinse well with water. After rinsing, drain the excess water with paper towels. The ninth step in our process is to again dip the slide 10 times in 95% ethanol with 3 changes. Last step in the H&E staining is to dip the slide 10 times in xylene with 3 changes. After the xylene process, wipe the edges and the back of the slide to clean it. Be cautious not to touch the tissue sections.
Good day everyone, my name is Jer Castaño and I will demonstrate the next procedure after staining, which is mounting stained tissue sections. So the purpose of mounting stained sections is to protect it from getting scratch or deterioration. Additionally, it preserves the slides for permanent keeping and facilitates easy handling and storage. So before we proceed to our procedure, let's start with the materials needed for mounting stained tissue sections. Using a dropper, apply a single drop of multi-medium upon tissue section. With the use of forceps, hold the cover slip at 45 degrees angle, allowing the drop to spread along the edge of the cover slip. Then, let go the cover slip and allow the mounting medium to spread slowly. After the mounting medium has spread to the edge of the cover slip, wipe around it for neatness. For the remaining stain slides, repeat the process until all stain slides are being mounted. If proper care has been taken, there should be no air bubbles present. If many are present, slide should be returned to the silene to remove the cover slip. Therefore, it will slip off and remounting should be done. Never pull the cover slip. And also, slight warming of the slide from below will make the small air bubbles escape from the side of the cover slip. If there are only few air bubbles present under the cover slip, applying gentle pressure on the slip may also do the trick.